Great. And Clint, thank you for introduction of Genus. So just to iterate, Genus is a genetic company. So what does it mean? We developing genetics through breeding as well as using precision breeding technology like gene editing. And we're working in the space of pigs, dairy, beef, and our daughter company CBS, that's our cattle company, bovine company, and PIC is a pig improvement company that is solely focused on improving genetics of pigs. So today I will talk about our gene editing efforts, and the main development is our PRRS-resistant pigs. But first is what PRRS? PRRS is porcine reproductive respiratory syndrome disease, and it's really appeared more than 30 years ago. And first it was detected in Europe, but very quickly it spread around the globe. And over all those years, there were multiple vaccines developed and continue to be developed for this very serious disease in pigs. However, we could not really manage well PRRS, and every year PRRS really brings significant losses to pig farmers around the globe. The virus is RNA virus, and the symptoms are very serious. Pigs start having fever, they develop pneumonia as a secondary signs post-PRRS, but it's really impact pregnant pigs significantly. You will have stillborn litters when moms get PRRS. And as a result, if pigs survive, they will have weakened immunity, and that leads to significant application of antibiotics on those farms if herd will even survive. So there is a solution to that problem, and that solution was identified by academic researchers at Missouri University and Roslin Institute. And it really all comes to one protein, CD163 protein. The protein itself is pretty benign. It's actually important protein for all mammals, and it's present in all mammals. The only difference that in pigs, it has very specific domain 5, that binds a CD163 protein and allows genome replication in the body of the pigs. And there is a very elegant solution, is to cut this domain 5 using CRISPR-Cas9 complex. And once that domain is gone, the rest of the protein continue to function and deliver on other function. But however, the protein cannot bind any more PRRS virus. So it's kind of looks like the door for the virus is closes, and pig don't experience any symptoms of the disease. And of course, it's the way we can do today, the gene editing, it's all done through protein complex with guide RNA. There is no foreign DNAs used during the process, and therefore there's no chances for foreign DNA to be introduced into the pig genome. Now, I will take you through this scheme that explains how we produce those pigs. So for us, it's very important that we use this approach because it's commercial. We're building commercial herd, and it all starts with fertilized zygotes that we're collecting from our donor gills. Then those zygotes get injected with CRISPR-Cas protein complex with guide RNA. And then we let the zygotes to culture to 4-cell stage. And at this stage, we transplanted back to the surrogate gills. Now, after gestation, the first generation of piglets, we call it E0, usually will have the right edits, but it also will have some edits that are not desired. It will have some mosaicism because of multiple alleles can be edited in different way. So this is the first generation of pigs that goes through very thorough molecular screening and selection, where we looking for only desired edits, and at the same time, we're selecting peaks that don't have off-target edits. Once we select those peaks, we cross them with wild type to really clean up all the mosaicism and from the genetic background. Once at that point, we create a population of E1 peaks. And again, they're all heterozygous. They carry only one allele. And again, we're going through extensive molecular screening, to confirm that desired edit present in all those peaks, and there are no off targets in any of those peaks. However, heterozygous peak cannot be resistant to PRRS because we need homozygous allele, meaning that we need to cut both alleles of this domain 5 from the gene and the resulting protein. So that's why at this point we're crossing those heterozygous E1 animals and to produce E2 population. And in this E2 population, we will have homozygous for edit peaks, heterozygous, and of course, wild type peaks. So we're confirming that the desired edit present in our heterozygous and homozygous peaks. And at this point, those are the peaks that we're taking into our, what we call building our commercial herd. And it's all done from that point on through conventional breeding. I think the one big challenge for us was to figure out how we can introduce exactly the same edit into all our breeding nucleus. And what does it mean? We have female and male lines in our breeding nucleus population. Those are the lines that we continuously working on improving for a variety of different traits, such as size of the litter, back fat, health, etc. And the crosses between those lines is really what produces a final product as a terminal peak. So we needed to introduce edits simultaneously into all these lines so that we could avoid any genetic bottleneck and prevent genetic improvements as we're working on breeding and providing final product to our customers. So that's the approach we took. We introduced exactly the same edit into each of these lines independently. And we continue breeding those lines now with edit, which really helps us to avoid and eliminate any bottleneck in our genetic diversity of our nucleus. Now, this E2 population is where we really started to look into a variety of different characteristics that are important for our regulatory application and approvals. And working very closely with FDA through all these years, we identify what study we needed to do to submit our package for FDA approval. And it so happened to be, it's a very similar package that we then can submit to other countries as well. So one very big difference, I think, with work with FDA, that we do need to confirm that our edit really confirms resistance to PRRS virus. So we've done a lot of live animal challenges with many different PRRS strains that actually today type 1 and type 2 that are today, or at least during last decade, are present in the field. And as you can see on the left here, that all our testing confirmed that edited peaks didn't have any infection with PRRS virus, and all non-edited peaks of the same genetic line were infected and showing signs of disease. Now, the next big part of the work we needed to do is to demonstrate that phenotypes of edited peaks are not different from phenotypes of non-edited peaks. So we looked at a variety of traits through birth, to finishing and also reproductive traits. And here you can see only key traits, but we have the tables and we publish them with a lot more traits. But as you can see here, at birth, we look probably most important birth weight. There were no difference, and we looked on both, on sire line and dam line, and we didn't see any difference. We also looked at weight at 140 days, lifetime, daily gain, carcass health, etc. And again, gene-edited pigs perform in every characteristics, phenotypical characteristics, exactly the same as non-edited pigs. And finally, we needed to look at meat composition because that's what really about the safety concerns or risks that we needed to address. And we evaluated meat quality, looking at carcass weight, muscle pH, marbling, etc., important characteristics for producers. And we also looked at the meat composition, if there are any differences that we can detect. And again, not in meat quality or meat composition, we did not detect absolutely no difference between edited and non-edited pig. So that work took us some time. Obviously, you need to produce statistically reliable measurements. So it does take quite a lot of pigs that we had to put through all these studies. And it takes time, right? Because again, we need to grow these pigs, we need to match them, and we need to have a reliable number of pigs for each of the study. So all the package of all the study, and of course, all molecular characterization data that I was describing to you, which we did at E0, E1, and E2, where we were looking at desired edit and off-targets, absence of off-target edits. That's the body of the work that comprised our regulatory packages. And when we're talking about regulatory, obviously, the first and most important for us is to get our approvals with the FDA. And that work, again, takes time. And we worked very, very closely with FDA. And really, I think we learned on both sides, on our side and FDA side, is again, the product of that size and was the first going through that type of regulatory approvals. We are very close at this point, and we still have some work to do. However, we needed to think about global pork trade, because getting approvals in US is not sufficient for us because pork is a global commodity, and we have to think about how we're not disrupting the global trade. So therefore, we needed to target countries that also produce or import substantial amounts of pork. We needed to think about countries that do not export pork, but might be relatively easy to commercialize, those countries like Colombia, China, or Japan. And obviously for us, we thinking about it as from the global perspective that we will be targeting widespread adoption of this technology. So that's really what played into our thinking about our regulatory strategy overall. So just to, again, talking about FDA, again, as I said before, this is really important for us to get our approvals in US. Fundamentally, that's really, without that approval, we cannot commercialize our pigs on a global scheme. So here what you see is different stages. I think Adam maybe was talking about it, but it's really convenient for producers' path working with FDA in a way that we can submit different parts of our application at different times as they're getting ready. So as you can see, we started submissions in 2021, where we talked about product definition, molecular characterizations, etc. In 22, we submitted full package on molecular characterization of 3 generations of our animals. And last year and this year, we finalizing all other study. As I was talking about phenotypic characterization, that's really extensive and takes time. We submitted and it's been accepted by FDA. Phenotypic durability and genotypic durability, it's all about how the trait and edit itself are inherited through the generations. So those studies done and submitted. Food safety studies also, and environmental safety that really addresses what will happen, for example, if our pigs get will get escape and can mix with the wild pigs population. So this is where we are today. Most of our technical submissions are finished. And right now we are working really on the post-commercial monitoring part, which means we need to report on regular basis about our pigs, edited pigs, any distribution to our customers, any incidents that we can observe, and of course, if there are any PRRS breakouts where we should not expect them and where we have all pigs homozygous for edit. And of course, it's our facilities inspection that we're preparing right now that will be important for commercialization of the pigs. At the same time, we also moved forward with our global approvals. As you probably heard yesterday already, we got positive determination in Colombia in 2023, as well as in Brazil in 24. And that means that in both countries, Colombia and Brazil, our gene-edited pigs would be treated as conventional and also go through all normal regulation that any conventional bred pig will go by securities, etc. We started our regulatory submissions in Canada and in dialogue with Canada regulators, and we're submitting our making our submission to Japan, and they're all happening in this month. Now, 2 important countries for us, China and Mexico. And in China, we are very hopeful. Regulatory environment is developing there, and they published regulation on gene-edited plants and animals. So we are working with our partners in China, and now we have permit to send our PRRS-resistant pigs for in-country assessment, and that's really exciting step forward for us. And Mexico is another country that we really need to get our either positive determination or approvals. And right now we are working closely with in Mexico to identify a path for opportunity to get such positive determination or approval in Mexico. As today, there is no gene editing regulatory framework that exists in Mexico besides the biosafety law that covers GMO. And with that, that's all what I have.